Well, I got a little cartoon in there. That's the best I can do. It's the, El Nino ENSO wave or sea surface height, animation. But I appreciate the opportunity to be here to talk about this subject. And I'm Tom Helser. I supervise and manage a life history population biology laboratory at the Alaska Fisheries Science Center in Seattle. And we've been involved in analyzing growth data for some time. We age a very large number of fish with using otoliths, and I think on an annual average we're aging probably about 50 or 60,000 otoliths. We've been doing this for about, I personally haven't, but the lab has been doing this for probably a half a century, and as I'll show in a little bit, we've got an archive of over 3 million pairs of otoliths that are warehoused at the Burke Museum in Seattle. So this talk is more statistical than biological. I'm not going to talk about any, underpinning metabolic or bioenergetic theory with regard to what we've observed in terms of variation in size with climate factors. Specifically, we're not looking at mean size at age as you would typically do in a large sort of composite data set. We're looking at individual growth variability as is displayed through growth increment measurements in otoliths. I know John Morangello's very familiar with this. He's published a very nice paper, I think, in Fish and Fisheries about the same time that we came out with a paper using Bayesian inference to apply this technology, I think in 2011. Is that my last caveat? I know I have a lot of them, but and by the way, yeah, this is actually a synthesis of a number of papers that have come out of our group. So not all the work is mine. I'm co-author on a number of them, but there again, I'm not the principal lead on some of this. Several people in my lab and collaborators have been working on this for some years now. So. Let's just sort of look at the type of data this is. This is in a statistical sense called longitudinal data. It's basically repeated measures on a subject over time, and in this case, it's a repeated measures of growth increment measurements from a time at birth to the time at death. And the high frequency variability in growth increment data is used to make inferences about the effects of climate variability on that organism. And this technique has been used in the tree science literature for many years. And in fact, most of the climate proxies that we have that span millennia are based on ice cores and tree ring data. So the paleoclimatologists will use multi-proxy data, which comprise ice cores, tree rings, corals, and a whole host of other things, and incorporate all those proxies together and kind of look at sort of how climate responded to these growth increment sizes throughout, several million years, if you can piece it back together that far. So what we see here is that the width of the ring is a function of some limiting factor on growth. It could be temperature. In the context of trees, it could be drought. Now, there's a whole host of things, and obviously the tree ring people have dialed this kind of tool in pretty well using cores that bore into the trees and they extract the samples. They're a lot more behaved than fish are. Fish are mobile, trees are not. And the ring widths in trees obviously are much more resolvable on an annual basis as opposed to otoliths, which actually present a little bit of a challenge because otoliths are actually highly variable, not only from an individual to an individual, the otolith increments, but even within a given sample, two analysts might actually come up with an observation error that could actually have a strong impact on the result. So we've been using this technology. Basically, I'm going to just step through sort of the basic idea of how tree ring scientists generate the relationships between climate and growth. And then I'm going to talk about basically sort of a more sophisticated approach, which is what we statistical folks like to do, to sort of incorporate everything in the model and use some information criteria to sort out what makes sense or what is robust or what has the weight of evidence. So this shows basically an otolith, a yellowfin sole otolith, and what you'll see is that there's an axis of measurement, and that basically use a micron or a software package like SigmaPlot or SigmaPlot. Sorry, it's SigmaPro or yeah, something like that. I don't use it much, but I'm a statistician, not a biologist for the most part. But anyways, you get these growth increment measurements. It's kind of odd because a lot of times people will, depending on the analyst, will choose an axis that is not consistent with somebody else. So again, there's another source of error that could be introduced into these analyses. Not so difficult for tree rings because they have a linear kind of identical scale in terms of the borer core that's brought out of the tree. From a fisheries perspective, this type of data actually has been analyzed quite a long time. And in fact, this longitudinal data is really following a single cohort. I mean, Christine looked at basically the mean size of a cohort through time. And so what she's doing is following this cohort, the mean size at age. And what we're doing here generally is following the cohort, but by the individual. And so if you get a random sample of individuals and you kind of model their individual growth variability from a mixed model context, you come up with a pretty decent expectation or prediction of how the growth is responding to different factors. But like I said, in the fisheries literature, it's been used for many number of years for a variety of reasons. A lot of times it was for back calculation of size or weight at age. And then more recently, Weisberg did some work looking at the statistical perspective of analyzing this longitudinal type data. He used a linear mixed effects model. And Strom basically used a GUI duct clam for long-term climate reconstructions. I'll talk about that in a little bit because we reanalyzed the data from GUI duct. And then Brian Black in 2009, Brian was basically a tree ring guy, and he saw the benefit or the utility of using otoliths and brought the biochronology concept to the marine system and started using calcium carbonate structures as a means of reconstructing or correlating growth with climate. And as I mentioned, John published, I think, that Fish and Fisheries paper in 2012. And ours came out about the same time. John's, by the way, was far more exhaustive in terms of its review. But anyways, the process of building a biochronology basically starts with, a group of animals that are collected at a particular site. And in fact, it could be across multiple species. And in fact, you might want to do it across multiple species because it provides more weight of evidence if you start to see sort of these functional responses. Because if more animals are responding the same way and in the same direction, it gives you more confidence that the signal is real versus sort of spurious. So basically, it starts with sample selection. You obviously want to get replicates by site and replicates by site within species. Again, you can do multiple species in multiple ecosystems. So you can understand we're starting to build a hierarchical structure with the data nested in each structure. The tree ring people develop these routines, one of which is a cross-stating routine, is basically to make sure that everybody's growth increment lines up relative to sort of some event that occurred in the past history. They use something called Koficha. And then finally, the goal is to detrend the data. That is by removing the intrinsic biological response over time. And that detrending essentially ends up to be a measurement time series. And you can see here's a whole bunch of animals with that detrended data showing high frequency variation over time. And finally, what they would do is get the weighted average of those individuals on a temporal scale, and then finally look at the correlation among, say, the master chronology, as they say, and sea surface temperature. So ultimately, they go through this process, and they get to the end point, and they got a time series that represents the population response, and then they correlate it to some sort of environmental factor. Well, I mean, you're kind of scratching your head, and I know you're going, well, boy, that might be kind of, you could generate a lot of spurious correlations that way because you're not propagating any error that exists in the actual time series measurements. And the only thing I want to mention here is that the temporal depth of the time series is generated by interlacing time series, so that if you want to build a time series that goes back 1,000 years, let's say, then really difficult for fish, by the way, but for trees, they have dead trees that are laying around in the same place that living trees are, and you core these and core them all, and basically you can interlace the time series and then work with a much longer time series. So we kind of perceive there's a lot of problems with that, and I think that the biggest is that it doesn't really, basically, the statistical properties of the master chronology or the average tree ring width are completely unknown, right? The true individual variability across subjects is not adequately characterized. And in theory, by doing that, you're actually estimating a number of parameters, but you don't know you are. You kind of think that, yeah, we have this time series and that's it, and that's the parameter that generated that time series. Well, not really, because you're doing this a bunch of individuals in a bunch of areas in a bunch of species, and then you've got this bloom of parameters for which you don't really know how to partition the variance. And so that's what we talk about here, is basically the uncertainty is not propagated, the error variance is not well estimated, if at all, and then there's other variance sources due to growth and climate effects that are not explicitly estimated, and in particular the environmental covariate. And then ultimately there's long-term effects of climate on growth that are ignored in favor of short-term year-to-year variability. So basically you might want to think about your time series as having some essentially non-stationary trend in the mean, or basically a step shift that you're not capturing. And so you might want to capture the mean response between one period of time and another time, as well as the variability in those two time steps. And we can do all that by an integrated modeling approach. And so, like I said, there's many statistical ways to do this. Linear mixed effects models or nonlinear mixed effects models, using a frequentist approach, using likelihood and REML, restricted maximum likelihood, or using Bayesian inference. And in this case, where we think of the data as actually inherently hierarchical. So it makes sense that we want to use some estimation technique that is able to, number one, explain the hierarchy of that data, the time series nature of it in terms of an autoregressive process, as well as and estimating all the error sources and then propagating those error sources in what's called a posterior prediction. And so, like I just talked about, the data are inherently hierarchical structured. We can estimate the hyperparameters, which is a way of collecting individual sets of parameters from a known distribution that we can estimate the variance from. Estimate the variance components is explicitly. There's some relationships to the outputs generated from this hierarchical modeling approach that are consistent with what the tree ring people use in terms of EPS, which is expressed population signal. And we can propagate uncertainty and evaluate model skill. It provides a hypothesis testing framework for different variance structures and the effects of environmental covariates. And like Uncle Fester here, who probably came up with the idea first, anyways, what that data looks is essentially like this, and it's kind of like a Walfer plot, where basically you difference the time series of otolith size at age, and what you have here is you get this measurement. You've got a time series of a time series that you actually scale to a common axis, which is age across all animals, right? In a population or even among different species. And so on the x-axis is the growth, the age at formation, and then the y-axis is the growth increment. It's a monotonically declining function. It's nonlinear, so what the heck? We'll use an exponential declining model. And so we have like multiple specimens of the same species in a given area, and then we might have another set of species in another area. Again, you can hierarchically structure this data any way you want, but you just need to recognize that it has hierarchical structure. And then basically we have a model that describes the mean response of that change for any individual over time. This is what's called the intrinsic growth response. Basically, it's just a combination of the genetic or phenotypic kind of responses that the animal has, and what you'd expect with this type of data. And so basically you've got, in this case, 3-parameter nonlinear exponential decline model, some error term there. And so each of these specimens can be described by a mean vector of the parameters k, alpha, and beta. Again, these are just to describe the intrinsic response. And these parameters we know are structured from a random perspective, where they are normally distributed with a mean of mu of those 3 parameters, and then a variance-covariance matrix which represents the random subject effect. Okay, and these are called the hyperparameters that we're going to estimate. So in terms of the model development, this growth increment data is basically individual or is completely described by this nonlinear intrinsic growth, biological growth with some error. And again, we've made some assumptions about the structure of these parameters, multivariate normal with a mean vector mu and variance covariance matrix. The error term is normal, mean 0, and sigma e for the error variance. And so at this point, we've described the individual random effect. Now we want to move into basically including more factors that will describe the existing variability as it exists in either the age dimension or in the year dimension. And so in one regard, you can decompose this error into now a climate effect. This could be, I like to think of sort of using the low frequency climate variability at this stage. In other words, for the animal we study a lot, there's the regime shift of 1967, or 76 and 77, and it was basically a pretty strong shift in the PDO. And what we have seen is that there's both a shift in the mean growth response in a non-stationary way, but also the variance structure between those two stanzas are different. So basically, I like to parameterize this low frequency effect first. And so I'm calling the parameter here as gamma, that is distributed normally with 0, and gamma y for the variance. And then at this point, you could add another effect in. Now, right here, we could call this temperature, like tau, all right? But for now, I'm going to just call it the effective year, okay? So we've explained some of the long-term or low-frequency variation. Now we're kind of focusing on the high-frequency variation. And we're going to call this tau, and this is basically, tau is distributed as a normally distributed a random variable with mean 0, and sigma h now as a variance. Sorry, I have a cold and I have to keep drinking. And so it just so happens that tau is really the master chronology, okay? And because the master chronology we saw was a mechanistic approach that removed the intrinsic growth and then just squeezed all the residuals, if you will, together from each individual into the weighted average. And so tau now represents a vector of random effect coefficients. So if you actually plot those random effect coefficients, you're going to get the master chronology. I've actually compared this to the tree ring master chronologies of many species, and they're almost identical. I would say that this actually has more variability because it actually allows for between subject variability to get incorporated into this high frequency variation. And the other thing is that sigma h is kind of an important thing here too, because if you take sigma h and divide it by sigma h plus sigma e, you get what's called the express population signal, and that describes the amount of variation that's attributed to the year-to-year variability. I should mention here again that you can then also decompose this other E into an autoregressive process. And I believe John actually talked about that in his paper from 2012. And so if you look at then, if you basically order the TAW, these are the random effect coefficients, you can see that it's this master chronology, if you want to call it that. I'm just going to call it the growth increment index. And from a Bayesian perspective, you can actually back out the marginal posterior distributions or densities of tau and put a confidence envelope around that time series. And I'm kind of an old guy, and I typically use old-fashioned programs like WinBugs, which is a Gibbs sampler using Monte Carlo Markov chain simulations. Okay, and so now I just want to show kind of how this all plays out. Is that if you had 8 individuals from this population, this is the increment growth data, and what you see, this dotted line is basically the intrinsic population response across all individuals. And if you stop there, you'd say, wow, that's really sucks for a fit. But then if you add if you look at then the individual responses, you can see that they're fitting the individuals much better. And then the other aspect of this is there's a property in this type of analysis called shrinkage, where basically data sets that are poorly informed are drawn towards the mean response. That could be an advantage at some point, particularly if you're modeling, say, von Bertalanffy growth parameters. And in some years, you might have poor data to inform or estimate the L max or K, and in which case this approach would kind of substitute some data in to try to provide a better fit to the data that's kind of non-informative. And if we add in next the year-to-year high frequency variation, basically you can see it's squiggling around, trying to hit the high points, trying to hit the low points. And what I haven't shown here is the incorporation of the low frequency signal, and that would even do a better job at kind of fitting this data. And again, this whole process requires a model building strategy where you can implement AIC or Bayesian information criteria to estimate basically what model structure, what covariant structures are most important. And right here, we're assuming that this is a symmetric sigma. The sigma is a symmetric distribution variance covariance matrix, but there's a whole host of different covariant structures that one might apply. And you can also introduce the covariant structure in the error terms as an autoregressive or a ARIMA process. Okay, so I'm going to start talking about some of the species we've analyzed. And again, this is not all my work. This is a synthesis of work that have been done. In some cases, it's not fully estimated using my approach from a Bayesian perspective. It might be used like R has the nonlinear random effects package, or some people have just, as one paper in here will show, just sort of that dendrochronology approach. They just look for correlations with the environmental conditions. I'm not sure if anybody described, I think maybe Christine did, but what we've got here is the North Pacific Current that hits about 48 degrees north latitude, and then splits basically between the California Current going south and the Alaska Coastal Current going to the north. And one of the species right here is gooey duck. It's a very good eating animal, produces a lot of meat. They're really hard to get to. You have to cram your whole body down a hole, basically, and grab it and pull it out. But it's a great animal to study because it lives about 200 years, right? And so you can basically extract the shell umbo and then do a peel and start measuring these increments. And I said, you know, like if you want a marine chronology, you want a pretty old species that has good annually resolved data, and geoduck is the cat's meow for sure. And so we have another species here, which is a northern rockfish. And northern rockfish are distributed actually both from California all the way to the Alaska coastal area. Black rockfish as well. And I'm going to talk about a study we did, or that I participated in, that used kind of the current systems as an experimental laboratory to understand the effect of the oceanographic factors and how they play out in terms of predictive growth. That's about it. This is a long-lived species. This is a medium-lived species. Northern rockfish lives to about 60, 70 years. Paul does the assessment on these guys in the Gulf. And black rockfish is a more coastal species that lives to about 20, 25 years. It's been pretty heavily overfished, and the size you see in the fishery of the surveys are much lower than what you'd typically see on a historic basis. And then I'm going to show some data that was analyzed from the Bering Sea and the Aleutian Islands. The Bering Sea is actually pretty complex. Kind of less complex than the Gulf of Alaska in the sense that the Gulf of Alaska has a lot of coastal structure and bays and fjord-type inner bays, and there's a lot of incoming kind of small rivers that are glacial-fed. A lot happens in the coastal area, actually, because it's a big nursery ground for just about all of the commercial species that are there. The Bering Sea is a super wide shelf. You have the Alaska coastal current coming through. This is the Alaska stream. Basically, it jets through the Aleutian Islands in this area. And then you got a slope current going this way to the north. You can see a temperature variation here. Again, this is during the survey period when the warmer temperatures are typically found near the coastal areas. It's consistent of three principal domains: an inner domain, which is near shore, the shallow; the middle domain, which is a more broad area, the shelf; and then the slope, which is a drop off to the abysmal plain in the Bering Sea. And the species we'll be looking at there's three flatfish that we worked on: summer flounder, Alaska plaice, and northern rock sole. These are the same critters that Christine analyzed in her database. Again, we analyzed northern rockfish from the Aleutians here, and also Pacific Ocean perch. And so, we're going to kind of develop a picture that a lot of the changes we see at the individual level are different, or perhaps more pronounced than what you see at the population level, like Christine's data showed. At the population level, there might be a lot more variability that gets imposed on the average size or weight at age from a large marine ecosystem perspective. Yeah. Is what?
You said summer flounder, but what's the top?
Yellowfin sole.
Yellowfin.
Did I say summer flounder? I used to work in the Northeast Atlantic, and I get my flatfish mixed up. Yeah. All right. So this is the earlier paper we did basically in 2012, and applying this method. This is a reanalysis of geoduck, my goal here was basically just to show that there were better methods to analyze this type of data. So what we did is we looked at geoduck in multiple places along the British Columbia coast. The temperature data came from gridded sea surface temperatures, which makes sense for geoduck because they're near shore, and sea surface temperature is kind of readily applicable and easy to derive. But and what you see up in this top graph is the predicted growth increment, and you see a credible interval. Now, the credible interval, as I said before, is just the posterior, the marginal posterior of the density function at that year. And you can see basically where the data is more informative, you can see that gray area, that gray area of the credibility inner shrinks quite a bit. And then overlying on that, you can see basically the Pacific Decadal Oscillation Index in terms of the climate anomaly, and also sea surface temperature. And what you'll notice there is that, you know, there's a good coherence between the increment growth, the predicted increment growth, and the PDO index, and even a better relationship between the increment growth and the sea surface temperature index. What I did actually is did it both ways. So I wanted to compare sort of, if I did the dendrochronology approach and I generated an R between, say, climate and the growth increment data, you know, I got a correlation of about, I don't know, 7, 8 or something like that. And then if I did it from a Bayesian perspective and I actually propagated all that uncertainty in 1,000 simulations and I looked at the marginal distribution of that correlation coefficient, it was actually about 0.6 or 0.658. So the important thing here is that, you know, what we see from mean responses and that sort of thing, where you're not accounting for a variability, I think we can go a little bit wrong on our inferences regarding what we think, or the effect to which climate is actually affecting the growth. And that's an area of concern that I have in general, without using the appropriate statistical tools. And so this shows this has actually been published since I generated this plot, but what I'm going to just show here, rather than getting into the nuts and bolts of the model and how the model applies to every single species, is just to show that there is some evidence, at least in the North Pacific where we are, that there's likely relationships between some climate factors and the growth in this longitudinal type data. And so this shows it for black rockfish. Again, the credible intervals get pretty wide on the ends of the data and then narrow in the middle area. And then, you know, you can look at the upwelling index, which is actually a dominant factor in basically the California current, and then the sea surface temperature anomaly as well. So I'll get to the synthesis of this in a second, and this isn't it, but this just shows a recent paper we did looking at intrinsic and environmental drivers in growth for rockfish. This is actually northern rockfish. And again, we used, I think Beth, who's in my group, Beth Mata, used a R using the nonlinear mixed effects package and generated these correlations between the PDO and the normalized growth index here on the lower part, and the sea surface temperature and the normalized growth index here. So it implies that, you know, there's something going on with regard to these climate factors affecting the growth of this species in this area. Another important point is that, and I don't really agree with the way this is generally done, but you can see that they typically